The difference between a pooled and arrayed screen can be tricky, so let's use humans as an example. Pooled screens are like grouping people in one room, whereas arrayed screens have each person in their own room. Pools are great for finding quick and easy differences, such as who wears glasses, but less obvious details, such as who has yellow socks on, would require a signal, like asking a person to raise their hand. Pooled screens using cells finds obvious differences, like cell death or cell survival, because missing cells and those growing out of control are easy to see. Less obvious things can only be found by marking the cells with certain things like antibodies or fluorescent reporters. Arrayed screens are great for less obvious things because each person is isolated, so it's easier to see differences such as who's wearing glasses. But unlike pooled screens, it's also perfect for finding multiple features, like who's wearing glasses, a sweater, and tie. If something happens only under certain conditions, like someone needing glasses when they read, this is like a cell expressing a specific gene only under certain conditions. Arrayed screens are done in multi-well plates, and there are a whole lot of them, while pooled screens are done in as little as one dish, and just like the name says, your results are pooled together. So a pooled CRISPR knockout experiment will have cells that are edited for one gene mixed with cells that are edited for other genes, whereas the arrayed screen has cells in each well with only one gene knocked out. The difference between one dish versus many tiny wells makes pooled screens easier to handle than arrayed screens, but you're limited to easy questions like cell growth and death. Automation, multiple readouts, and complex data analysis is possible for arrayed screens. And there's more choice because we have both lentiviral and synthetic guide RNAs. And our cherry pick option is perfect for arrayed custom libraries. Coming soon, we'll have custom pooled libraries for CRISPR knockout and CRISPR activation or interference available too. In the end, it's the biology of your cells and the question you want to ask, which will decide which screening method to choose for your own work.